Team 7, my name is Sofia and today I'm talking about cloning. Cloning is the science of creating a genetic copy of an organism. Sounds futuristic, but it's real. In 1996, scientists cloned Dolly the sheep from an adult cell, proving this could be done. There are two main types. The reproductive cloning, that creates a whole new organism. This could help endangered species, but it's still debated. And then we have the therapeutic cloning, that creates stem cells to treat diseases like Parkinson's disease, but it's also controversial. And here's a recent claim. In 2025, South Korean scientists cloned a Siberian tiger using a lioness and a surrogate. We still don't know if it's real or fake. Thanks for watching.
Hi, my name is Elena Villarreal and I'm going to talk about bringing extinct animals back to life. Well, in 2025, a company called Colossal Bioscience announced the birth of 3 genetically modified wolf pups, and these pups have traits of a long extinct wolf. Scientists used ancient DNA to make 20 gene edits to modern gray wolves by making them stronger and bulkier, just like their prehistoric cousins. And there are 2 key concepts that we need to understand for using in this context, which is the functional de-extinction, which means they are not bringing back the exactly species, but they are recreating them in modern animals. And the gene editing, which is a powerful tool that lets scientists edit DNA with incredible precision, allowing them to insert the ancient genes into living species like modern animals right now. And there's also a new, in early this year, scientists also recreate a woolly mice that is a tiny mice with really thick fur, like mammoth fur. And this test run for bringing back the woolly mammoth. And it actually worked because the mice develop a cold resistance, proving that the ancient traits can be revived.
Hi, my name is Alexia Feliciano and today I'm going to explain the stem cells applications. First, the stem cells can develop into different types of cells in the body. There are 2 main types that are the embryonic stem cells and the adult stem cells. Some most common uses to the stem cells are the bone marrow transplants. Also, they are used in the regenerative medicine. They are also used in the drug testing and disease research. Even though there are still challenges, the stem cell research has made very big progress, and in the future it could lead to new treatments and for many serious conditions. And that's it. Thanks.
Transgenic foods, also known as genetically modified GM foods, are produced from organisms whose DNA has been altered using genetic engineering techniques. This process often involves transferring genes between different species to impart specific traits like pest resistance or improved nutritional value. A fun fact about GMs is that they can actually be used to remove allergens from food. Scientists in the United States and Britain are already working on GM products such as safer peanuts and gluten-free wheat. The first crops developed were used for animal or human food and provide resistance to certain pests, diseases, environmental conditions, spoilage, or chemical treatments. The GM crops currently on the market are mainly aimed at an increased level of crop protection through the introduction of resistance against plant diseases caused by insects or viruses, or through increased tolerance towards herbicides. Scientists have recently gene-edited bananas to prevent them from browning. They were made so that they could cut food waste, and scientists are planning to launch them later this year.
Hi, my name is Regina Torres and today I'm going to be telling you about the gene therapy. So first I'm going to start with a fun fact. The first approved gene therapy treatment in humans happened in 1990 and it was used to help a girl with a rare immune disorder called ADASCID, which is a recessive genetic disease affecting the immune system. Now I'm gonna tell you two key concepts about gene therapy. The first one is viral vector. This is a harmless virus that is used to deliver healthy genes into the patient's cells. This is practically a virus that is bringing new instructions to the human body. And then we have gene editing, that is thanks to tools like this one. We can now precisely fix mutations in the DNA, almost like using a genetic spell checker. And finally, a recent new highlight is that in 2023, the FDA approved the first SRASPR based gene therapy to treat sickle cell disease. Sickle cell disease, a painful genetic condition that mainly affects people of the African descent, and this is considered a huge step forward into personalized medicine. And that's it for gene therapy. Thank you.
Hello, my name is Jimena Mabeli Vázquez Tamayo, and I'm going to talk about limb regeneration. First, talking about regeneration, it generally means regrowth. Us, as humans, we generally have a limited capacity or ability to regenerate organs, such as liver. Now, talking about the concept of limb regeneration. This is the ability to regenerate complex structures and reconstruct entire body parts from damaged tissues. It is mainly found among invertebrates such as amphibians. Now, an amazing fact. The CRM team has developed a new therapeutic treatment to regenerate these damaged tissues using bone marrow mononuclear cells that are extracted from the own patient bone marrow. These cells are programmed to fight inflammation and also to help newborn cells to grow. We are Team 7. Thank you for watching.